Hey YouTube, and welcome back to Science with Sully. If you like the channel, be sure to give the video a like, a follow, and if you really like the content, subscribe as more content will follow. And we all want more science content. In today's video, I will be going over the process of exocytosis and its mechanism. First, we need to do some housekeeping and talk about exocytosis for a second before we get into the exact mechanism. Exocytosis is the moving of substances from the inside of the cell to the outside. It could be thought of. This can be thought of as secretion. The cell is secreting a substance or proteins, maybe for other cells that are nearby, or maybe it just needs it out of the cell. There are plenty of reasons for exocytosis. This is a specific mechanism. The cell is very specific on what it is dumping out into the extracellular environment. This is very, very regulated. And like I always like to do with processes, let's break down the word into its prefix and suffix. Exo is Latin for exit, and cytosis, as we have talked about before, is in reference to the cell. So substances are exiting the cell. And that's a great way to remember exocytosis once we talk about the mechanism. So let's talk about the mechanism of exocytosis. First, we're going to draw our bilayered cell membrane. And let's label that so we know that. This is our cell membrane. And this would be the outside of the cell, or the extracellular matrix, and this would be the inside of the cell, or the cytosol. So somewhere downstream here, we would have our Golgi, and the Golgi has taken something maybe from a protein from the cytosol or from the endoplasmic reticulum, and the Golgi is basically saying it's time to get this substance out. So this will be our substance right here. We're trying to get that out of the cell. So what the Golgi is also a bilayer, like our plasma membrane, and the Golgi is basically going to create a vesicle and pinch off membrane to where it will be a bilayered. That's not a very good one. Let's draw that vesicle again. A bilayered vesicle, and we're trying to get this out of our cell. So that vesicle will move along our filamentous network, and it will come up to the plasma membrane, and it will get really, really close to the plasma membrane, and the membranes of the vesicle, which let's note that that is a vesicle. The membranes of the vesicle is the same membrane as the plasma membrane. So these membranes will fuse together and kind of dissolve into each other to make one membrane, as we are about to see. So they're going to push together, and there are proteins that assist in this. These would be VAMPs and SNAP proteins. But after they push together a lot, they are going to fuse and create a pit, and our substance that we've been trying to get out, this that we denoted as our red square, is now free to leave the cell and go to the extracellular matrix. And at the extracellular matrix, this could be This could go to another cell. It could also it could be destroyed. There's plenty of fates for this substance. But what the big picture was is that we want it to exit the cell, and the process for that is exocytosis. And remember, exocytosis is specific. We do not want to be dumping out our machinery that we need to carry out our cellular functions. So this is a highly regulated, highly specific process, unlike pinocytosis, for example. And in case you need to know these proteins, the proteins on the vesicle that bring it to the plasma membrane are called VAMPs. And on the membrane are called SNAPs. And they just fuse the membrane of the vesicle to the cell membrane, and that creates our pit right here. So if you like the video, be sure to give it a like, the channel a follow, and if you really like it, subscribe for more content that's always coming out. And I appreciate you stopping by for Science with Sully.